welcome to North American Food Explorers virtual conference, Food Forward, Growing for Tomorrow. Thank you for joining us. My name is Eric Bina, and I'm a volunteer member of the North American Food Explorers, NAFEX for short. I'll be serving as the facilitator for this session, which is Pears for Your Heirs. Before we introduce today's panel, a few items about Zoom. This is a webinar format. Participants' audio and video are automatically disabled. We do encourage you to ask questions. Please use the question and answer tab at the bottom middle of your Zoom window. If you're new to Zoom, there are some adjustments you can make to your view on your device. And finally, this webinar is being recorded and will be made available to members and conference participants at NAFEX.org. So if this is your first session, let me say a few words about NAFEX. We were founded in 1967 as a network of individuals throughout the United States and Canada devoted to the discovery, cultivation, and appreciation of superior varieties of fruits and nuts. NAFEX members share ideas, information, experiences, and fruit propagating material via our website, social media channels, fruit-specific interest group meetings, and annual conferences like this one. As a paid NAFEX member, you get 4 editions of the POMONA Journal each year, as well as the ability to search 50 years of POMONAs in our digital library, which contains a wealth of fruit growing information. This organization exists because of fruit growing members like you, and we encourage you to continue your membership and become actively involved as an interest group member, committee member, or board member. Please visit our website to learn more at nafex.org. Now let me introduce the panel members who will be speaking today. We have Nahla Bassil from the USDA National Clonal Germplasm Repository, Joseph Postman, now retired from the USDA National Clonal Germplasm Repository, and Marcus Toole, a home orchardist from Georgia. Before we start Pears for Your Heirs, I want to have a brief statement about my love-hate relationship with this saying. It was an old pear tree, probably planted 30 years before I was born in my childhood neighborhood, which gave me my first experience of delicious fruit picked from the tree. So I love the idea that someone planted that tree so I could enjoy that. But later, when I wanted to plant fruit trees of my own, the idea of pears for my heirs upset me. I want pears for me, not for some unknown heirs. Fortunately, wiser heads convinced me that I can, in fact, grow pears and get fruit from them. So I learned to graft, and I now have that childhood pear tree growing in my current yard. And every fall, when I pluck a ripe fruit and bite into it, for a moment, I get to revisit that childhood wonder of discovery of fresh fruit off the tree. So this is the magic of fruit trees for you and your heirs. Now, let's get started. It is my pleasure to introduce Nahla Bassil. Dr. Nahla Victor Bassil is a plant geneticist with the U.S. Department of Agriculture's Agricultural Research Service and National Clonal Germplasm Repository in Corvallis, Oregon. She received her bachelor's degree from the American University of Beirut and her master's and PhD at Oregon State University. Since starting her position at the National Clonal Germplasm Repository in 2002, her primary research focus has been DNA marker development and its applications to germplasm management and diversity evaluation in plants, with an emphasis on temperate fruits and nuts. So Nahla, take it away.
Thank you very much for the introduction, Eric. Joseph is going to start our presentation and I will be jumping in during the presentation.
Yeah, so Eric, I think both Nahla and I will be unmuted, and I'll take the first few slides and then turn this over to Nahla. So I don't know if you're planning on introducing me afterwards or if you want to introduce me now, and we'll sort of tag team it.
I'm willing to go with your introduction now if you want. Joseph Postman, retired in August 2019 from the U.S. Department of Agriculture's National Clonal Germplasm Repository in Corvallis, Oregon, where he worked for more than 35 years as a plant pathologist and curator. He helped to develop the Living World Pear Collection, representing genetic diversity of pear cultivars and species from around the world. Along with his wife, he is now tending his personal collection of 100 or so apple varieties, an assortment of blueberries, grapes, and pears, and a flock of Wensleydale sheep on their small farm outside of Corvallis. Okay, Joseph, you start then.
Okay, great. Thank you, Eric. And let's see, before I bring up my PowerPoint, the way we're going to handle this today, I've got a few introductory slides, and I'll introduce the Germplasm Repository in Corvallis and the new curator who was just hired to replace me. And since we're going to be talking about some technical techniques for evaluating pear genetics. We'll turn it over to Nahla to introduce those techniques, and then I'll share some of the results that we've obtained. So let me share my screen here and get this going. All right, I think you should be seeing the title slide, and let me make some adjustments so I'm not looking at myself. Good. So the title of this session is Pears for Your Heirs. And thinking about that, I think we're going to be talking a little bit about the heirs of our pears, because part of this talk will deal with some of the ancestors, some of the parents that led to some of our modern cultivars. So Chris Homonix, when he invited me to give this talk, was aware that we were using some various DNA techniques at the gene bank in Corvallis. And he was wondering how those played into the management of the collection and some of the things that we've discovered. So let's see, we've put together, and I'm gonna have to, this problem yesterday, getting to the point where I can, there we go. Here's an outline of our talk. I'll do a little introduction to the USDA Gene Bank and introduce the new curator, Lori Reinhold. And then just mention some of the various challenges that we have in managing large plant collections. This is something that affects us at the Gene Bank in Corvallis and every one of you that has large collections of fruit trees. Then we'll turn it over to Nahla to introduce especially two of the DNA tools that have become common for evaluating collections, whether it doesn't have to be plants, it can be plants and animals. The techniques are pretty much the same. So then I can refer back to those when I talk about some of the pedigree information that we've discovered and some of the redundancy information. So first, a couple of words about the USDA National Plant Germplasm System. Many of you are aware that there are a series of gene banks scattered around the United States. The Gene Bank for Pears and Hazelnuts and Berry Crops is located here on the West Coast in Corvallis, Oregon. Those of you working with apples are familiar with the Apple Gene Bank in Geneva, New York. The Davis, California repository maintains quite a few different crops, prunus, grapes, walnuts, pistachios, almonds, a number of other things. The citrus collection is in Riverside, California, pecans in Texas, and then a number of tropical crops are in 3 different stations in Miami, Puerto Rico, and Hawaii. In Corvallis is where the World Pear Collection is located. On the right there, you see Lori Reinhold, who just started less than 2 months ago. So she wasn't really in a position to be giving this presentation on the pear collection. She's just getting her feet wet on how to survive in a USDA facility. But when I left in 2019, the pear collection had over 2,000 accessions of growing pear trees in field collections, and 300 or 400 seed lots representing mostly wild relative species. The wild relatives aren't only kept as seed, but we do have samples of each of the wild pears as growing trees to make them available and to allow them to be used in evaluation research. But the numbers on the left there summarize the collection in terms of cultivars. There's over 1,000 European cultivars, over 200 Asian cultivars, a little over 100 hybrid cultivars. And by hybrid, I mean they're hybrids between a European pear and an Asian pear. Those of you who live in the South are very familiar with hybrid cultivars. There's quite a few selections that have been made as potential rootstocks. We still don't have very good clonal rootstocks for pears. There are a handful available. And in the last few years, we've seen an explosion of interest in peri pears or cider pears. I don't think we're going to be talking any more about those, but if you have any questions, we can address those in the question and answer session. In one of Monday's sessions, a question came up about GMO apples and pears, and so I threw in this slide. I wanted to clarify for you folks who are not familiar with the genetics and genomic techniques. I wanted to distinguish between genotyping, where you're using techniques to read DNA, and that's what we're talking about today. And then there's other techniques that are used for modifying DNA. So none of the plants, I'm not aware of any apples or pears that are genetically modified, except for a single group of apples that were developed in Canada. They call them the Arctic series, where they've had a gene modified that silences an enzyme that causes browning when the fruit is cut. So these few varieties of genetically modified apples are primarily used for marketing prepackaged sliced apples, and they don't brown when these Arctic apples are used. But the tools that we're talking about today are DNA reading tools or DNA evaluation tools, and they will help us with some things that in the past we had to do by researching historic books that gave us descriptions and illustrations of different varieties to help us document redundancy, to verify cultivar identity, working with taxonomists to verify the taxonomic identity. Molecular geneticists are now characterizing different species of pears. We're primarily going to be talking about cultivars today. Some other things that DNA tools can help us with is to assess the genetic diversity of our collections and see if we've got an overabundance of plants that represent a narrow group of, say, European pears versus the world's, the pears that are available around the world. We also are now able to use DNA tools to validate the pedigrees and also to reveal the unknown pedigrees of some ancient cultivars that we had no way of knowing before. And we'll share some results of that information with you today. Breeders are finding DNA tools very, they make plant breeding much more efficient because now we have markers for some traits so that you can screen a population of seedlings before they're planted out and only plant out the plants that have some of the traits that you're looking for, such as disease resistance or ripening traits or pollen compatibility. So I'm going to turn this over to Nahla now, and Nahla, you're going to have to tell me to advance the slides to talk about primarily two DNA tools that we use for evaluating germplasm collections. So take it away, Nahla.
Thank you, Joseph. I'm very happy to be here and give a very brief overview of the DNA tools we use. And so two of the most common DNA tools we use are named simple sequence repeats and single nucleotide polymorphisms. In this slide, I try to explain what they are. So in this top panel, do you see my my arrow or not?
No, I think I'm going to have to do the arrow. So
yeah, so this top panel, what you are looking here at now is an alignment of the DNA sequence in the same location on the chromosomes between cultivars Bartlett and Old Home. So what you see, so if you see like the line between the bases here on top, A and A, it means that they have the exact same sequence. So in this box, you basically are seeing that Bartlett and Old Home have the exact same sequence and at this region in the genome, except in the region where there is a yellow box. So if we look closely at this yellow box, you see in the red box a sequence CCTCC. This is a repeat sequence of 5 base pair. It's a simple sequence repeat, which is defined as a 2 to 7 base pair DNA sequence that is repeated in tandem right after the other. So here in Bartlett, you see that we have 5 repeats of this CCTCC, but in Old Home, you see that 3 of them are missing. So they are not there. So so this is a characteristic difference between Bartlett and Old Home at this location. So if we are to amplify this region and we amplify it by designing small pieces of DNA forward where the blue is, it's a forward primer and a reverse primal that allows us to start the DNA amplification in that region. We use something called polymerase chain reaction, which is just an enzyme that we add to the DNA of pair of Bartlett and Old Home that we're trying to amplify and trying to genotype. So we basically make a lot of copies of this sequence, and then we separate it by size, and then we can see that Old Home, the fragment that is amplified is shorter than the one that is amplified in Bartlett. So so in human diagnostics, they use that technique for parentage analysis, for fingerprinting, and we use exactly the same technique in plants. We use what we call SSR-based fingerprinting to look at the genotype, to fingerprint our cultivars and see if they are what they're supposed to be. Another type of marker, single nucleotide repeat, a polymorphism repeat is, so here what you're seeing is in Bartlett and Oldham, a DNA sequence at the same exact location. So you see that the sequences are identical except for a single base. And that's the one that is, there is a circle there in blue. So in Bartlett, you have a TA instead of the CG in Oldham. So you have a single base that is different between them in this location. And when we genotype them at that location, we want to see who has the TA and who has the CG and who has a combination of the two. Next slide, Joseph. So to develop these marker techniques, we have to have the sequence information, and there's been a lot of advances in the last 10 years or so that allow us to do a lot of DNA sequencing. And then we use software to identify these regions, either SNPs or microsatellites. Then we design either small DNA sequences that are called primers or probes using another software. And then we apply that to the extracted DNA of the sample to genotype them. So in genotyping, on the left, you see an SSR-based fingerprint of pear. So this is at 11 of these different locations. So it's called an 11 SSR fingerprinting set because it assays the DNA at 11 locations on the chromosome. And at each location, we are able to tell what are the sizes that are amplified. So if you look at the first one in blue, those two peaks, you see that in GEM, we get a 94 base pair and a 97 base pair allele or fragment size in GEM. And that is characteristic of GEM. And we look at that at all 11 of these locations. So that's SSR-based fingerprinting. On the right, you see a result from fingerprinting or genotyping pear at 70,000 SNP locations, 70,000 locations in the pear genome. Each dot here is a single, each dot is one cultivar, is the genotype of one cultivar. Depending on where they fall in this cluster, we know what the genotype is. So the blue cluster in triangle, it has the BB allele. It has the genotype there. It's homozygous for one of the alleles. It could be, we know what it is. We call it here BB. All of the yellow dots are heterozygous, or they have two different alleles. And then all the red ones are homozygous for the other allele. So at the SNP location, you will have one or the other base. Since we have two different copies, pear is diploid. It gets a copy from the mom and a copy from the dad. So you're looking for the composition at this location will be two alleles. So either the parents have different alleles, A or B, and then they give A, B to the progeny, so they'll be heterozygous, yellow, or they have the same copies. Anyway, so it's a different technique, and we have different ways of assaying for the composition at that location. And then we have to do a lot of data analysis to be able to answer the questions that we have. Next, Joseph. So here is how we interpret the data in Old Home by Farmingdale. So Joseph will tell you later about the Old Home by Farmingdale rootstocks. So what you're seeing in this table is the genotype at 3 of these different SSRs. So on the top, you have the name, you have EMPC117, 2 of them next to each other. So this is one of the SSRs. So you basically have 3 of these locations, and you have the genotype of Old Home at each of these 3 locations. And same with the Old Home by Farmingdale, Bartlett, and Farmingdale. So Old Home by Farmingdale was believed to be the result of a cross between the mom, Old Home, and the dad is supposed to be Farmingdale. If you look at the DNA fragments that are amplified in Old Home at EMPC117, they're not 119. And then in Farmingdale, it's in red, 99 and 121. So if Old Home by Farmingdale is the result of the cross between the mom, Old Home, and Farmingdale, then they should have, you know, one of the alleles from the mom, 119, which they do. Both Old Home by Farmingdale 333 have the 119 from the mom, but they do not have 99 or 121 from the dad. So then we know that Farmingdale is not the dad. And we look at that at multiple locations on the chromosome. And so you can see that Bartlett has given, you know, Bartlett has 117, and both Old Home by Farmingdale rootstocks have the 117 allele. So when we look at the composition of, or the alleles of these SSRs at each of the location, then we can tell who's the real mom and dad and who is not. So then in this case, we know that Old Home is the female parent, Farmingdale is not the male parent, but Bartlett is. Okay, next, Joseph. So this is a very complicated slide, but I'll make it simple by explaining it. This is an experiment we're doing to develop a DNA test that can tell if a seedling is resistant to fire blight or not. And we have to do a lot of research to get to that point. We've done a lot of the research and we're close to the end now. So the research is first to find the chromosomal region on the pear chromosome that controls fire blight resistance. So what we did is we worked with a breeder, Richard Bell, in Kearneysville. He's retired now. He had 3 families, large families, about 100 trees each, that were derived from a cross between a susceptible and a resistant pear. So in this case, Joseph is showing you the families. Those are the 3 families. So in the first family, Old Home by Bartlett, Old Home is the resistant and Bartlett is the susceptible. So for each family, you have a resistant and susceptible parent. So what we do is we genotype the entire family, all these 3 families, with the SNP array, the 70,000 SNPs that have these markers, and we're able to find, and then we look at the phenotype too, which one of these is resistant, which one is susceptible. Then we're able to find the region on all the chromosomes of pear that control fire blight resistance. So here you are seeing a diagram showing all the markers on chromosome 2 of pear, and you see a bar from 2017 and 2018 that highlights the region that was found to be responsible or controlling fire blight resistance in pear. So then what we do with the new sequencing technology, what we did is we sequence the genes in those regions from the susceptible and the resistant parents. And then we use statistical analysis to identify, you know, bases that are associated with resistance. And so right now we are developing a genotyping assay to for all of these regions, which were about 531 bases. And then we are genotyping them in close to 500 pear trees for which we know whether they are resistant or susceptible. And we're trying to develop a test to see if we can diagnose fire blight resistance. So this is all the fun stuff, a lot of the fun stuff we do with DNA tests. They can be very useful, and Joseph will continue next, explaining what we have discovered in terms of pedigrees and redundancy in pear.
All right, well, thank you very much, Nahla. And so I may or may not just refer to the terms SSRs or SNPs when I'm talking about the techniques that were used to determine this information, but Nahla introduced this story about Old Home and Farmingdale, which are the parents of a series of rootstocks. And these two parents were selected based on the work of Frank Reimer, who was the superintendent at the Oregon State Experiment Station down in Medford, Oregon, earlier in the 20th century. I think he was there from the early 1900s through about 1950s. And fire blight was a big problem for pears, and one of the solutions to fire blight was to use a fire blight resistant rootstock or a fire blight resistant trunk stock, so that if a tree became infected with fire blight, it wouldn't move all the way down through the tree, it would just kill the branches on one side or another. So Reimer ended up visiting Benjamin Buckman, who was a plant breeder and collector in Illinois. And Buckman had a few varieties that he claimed were very resistant to fire blight. One of those he called Farmingdale. Buckman lived in Farmingdale, Illinois, and so that's where that cultivar got its name. Another was Old Home. Old Home was a variety. You can see them pictured up on top here. Old Home has almost round, I would say almost worthless fruit. It's almost inedible. Farmingdale was a seedling that grew up in the vicinity of an anjo tree. So Buckman felt that it was probably a seedling of anjo. And after many, many epidemics of fire blight in Illinois, these two varieties stood up as being very resistant to fire blight. So Reimer brought those back to Oregon, and he used those for a lot of his experiments, testing them with fire blight, making crosses with them. And he found that they were some of the most resistant pears among the hundreds of varieties that he had in his collection that were resistant to fire blight. Reimer went on to, well, he didn't actually make the Old Home by Farmingdale crosses, but a nurseryman took Old Home trees and planted them up in British Columbia in the vicinity of some Farmingdale trees, assuming that those two trees would cross each other. Then he collected seedlings and grew them out from these Old Home open pollinated seedlings, presumably with Farmingdale as the pollen parent. And these are some of the remnant Old Home by Farmingdale selections that are at the Germplasm repository today. They had over 200 seedlings that were screened at Oregon State University for ease of propagation, for resistance to a number of different diseases. And then when Nahla was developing these SSR tools for fingerprinting the collection, we thought we were making some kind of error. I've got a tree here, part of a dendrogram that shows Old Home here that grouped very nicely with some of the Old Home by Farmingdale. We used 5 different Old Home by Farmingdale selections in this initial work. Old Home by Farmingdale 230, 51, 333, and a few others. But we thought we were doing something wrong because Farmingdale never grouped with any of the Old Home by Farmingdale selections. It did group with anjo, suggesting that Buckman was right. It was probably a seedling of anjo, or at least related to anjo. So we wrote a paper reporting that this was probably not the father. Farmingdale was probably not the father of the Old Home by Farmingdale rootstocks. Here's an image from that paper showing Old Home, showing Farmingdale, and showing Bartlett, which we found just by accident because Bartlett was included in a control in that SSR study. And here are 3 different Old Home by Farmingdale rootstock clones. Normally you don't see the fruit on these because these are sold for the rootstock, but they do make fruit, and some of the fruit is better than others. This is the fruit of Old Home by Farmingdale 51, and you can see that it just physically resembles Bartlett and more than it does Farmingdale. Likewise, Old Home by Farmingdale 87, it's more round like Old Home. And 333, which is a rootstock that a lot of you might be seeing if you're ordering semi-dwarf pear trees from a nursery. Now, more recently, we have this SNP technique available to us. As Nahla said, that looks at 70,000 different positions on the genome where the plants either match or they don't match. And that's a very strong relationship. So in this study, we were able to look at all of the Old Home by Farmingdale selections in the collection. So here's all 41 Old Home by Farmingdale selections with the pedigrees that Sarah Montanari was able to generate. And you can see for almost all of them, Old Home and Bartlett were the two parents. There were two that had Anjo as the pollen parent and not Bartlett. So this is some of the pedigree information that we're able to generate with some of these new DNA sequencing tools. Some other interesting results that come out of this pedigree analysis, Sarah Montanari generated SNP data for the entire pear germplasm collection, which doesn't have every single European cultivar represented, but it does have quite a few. So some of the very old cultivars like Anjo and Bartlett and Comise, which we have no way of knowing what their original parents were, if we happen to have the two parents in the germplasm collection, Sarah was able to determine that the actual parents of Anjo are an old variety Sucre Vert and White Doyenne. Likewise, Bartlett has White Doyenne as a parent. Comise is a hybrid between an old cultivar Gulot Marceau and Duchess d'Angoulême. Farmingdale, we in the SSR work, we were able to see that Anjo was likely one of the parents. Well, the SNP analysis shows us that the actual parentage of Farmingdale is Anjo by Duchess d'Angoulême. Some of you in the Pacific Northwest are familiar with some cultivars like Orchis and Rescue, which have done very well under Pacific Northwest environments. These were just seedlings that were discovered in the wild, and we weren't sure whether they were escaped old European cultivars that were from an old homestead or whether they were seedlings that originated in the northwest part of the United States. Well, now we can see that the Orchis pear is actually a seedling of Bartlett with Doyenne de Comise as the pollen parent. And Rescue is a hybrid of the winter pear Bear Clergot with the summer pear Clap Favorite, two very nice quality pears. Seckel has been around for almost 300 years, and now 300 years later, we were able to determine that the parentage of Seckel is a White Doyenne and Rousselet de Reims. I'm going to be talking a little bit later about some of the hybrid cultivars that you're familiar with in the South. Baldwin is one that we now see that LeConte is one parent. We don't have the other parent in the collection. Kiefer is thought to be a parent between a Japanese pear and Bartlett, and the DNA evidence bears that out. Bartlett is one parent, but we don't have the Asian pear that was the other parent. It's also fun. You can go into a grocery store and buy a Chinese pear in an Asian market. I grew a number of seedlings of Ya Li, and they were included in this SNP analysis. And sure enough, Ya Li that we took the seeds from is one parent as expected, but we happen to have the pollen parent, which was another Chinese pear. So we know what that farmer in China had to pollinate his crops. You'll see that a number of these European pears that are high quality pears share a common parentage, and breeders call those founder parents. If there's a common parent in a lot of different cultivars, whether they're European or Asian cultivars, a founder cultivar is a founder, is a common parent that is a high quality and was used either accidentally or intentionally in a number of crosses. And looking to see in a collection if a lot of pears have the same founder parent, that shows that you've got relatively narrow genetic diversity, whereas if the collection has a lot of different parentage, that shows that a collection has more broader genetic diversity represented. Here's an illustration on the right of White Doyenne. So I've pulled out some of the pears that we call these modern founder cultivars because Bartlett, Comise, Seckel, and Anjo turns out are the parents of a lot of, whether they're sports or whether they're seedlings that are derived from these. I don't list all the cultivar names, but Bartlett, there are a lot of sports in the collection. There's red Bartlett, there's russet Bartlett, there's Bartlett that's more or less compatible with quince as a rootstock. I'm going to speed up here because I don't want to cut into Marcus's time. Like also the ancient European cultivars, and I was going to mention about the modern cultivars. We call these modern cultivars, but the cultivars that you see in the grocery store today, these all originated more than 200 years ago. In the case of Seckel and Bartlett, it's more like 300 years ago. But in this SNP analysis, we were able to see that White Doyenne is a founder cultivar for quite a few pears that are in the collection. Gros Morceau is also a founder cultivar. It's the parent of 43 different accessions, and it's also many of the accessions that are seedlings of Gros Morceau are also parents of other accessions. So there's many generations of parentage. Rousselet de Reims, which is one of the oldest cultivars in the collection, it originated at least 500 years ago, and it might date back to Roman times. This is in the parentage of more than five generations of offspring in the pear germplasm collection. Duchess de Angoulême is another. I'm going to take a quick break here. Eric, if anybody has any questions about the technical stuff that Nahla shared or the pedigree information, maybe this is a good time for one or two questions, and then I'll talk about some of the redundancy that we were able to determine in the hybrid cultivars. Have you gotten any questions?
We have one question here. It says in the genetic, this is from Chris Heater, in the genetic areas identified to be associated with being resistant and susceptible, what kind of genes are found in those areas?
Okay.
I assume you're talking about the marker genes for fire blight. Okay,
I assume so, yes.
Yeah. So, in that region, I think there were around maybe 200 genes or so. There are genes that are known to be to control disease resistance. They have a certain structure that we know of. They're called NBS LRR genes. They have a leucine-rich repeat. They have a region that binds. So we know what the composition is. So they're disease resistance genes is the short answer. But we don't know if we don't know which one it controls fire blight yet. You know? Yeah.
And for the listeners, I want to encourage people to ask questions. I know there's been a lot of technical information here, but even if you don't have a technical question, if you have more general questions about pears, we hope to be able to field some of those as well. Okay,
Yes.
Justin.
All right, well, I'm gonna launch back in because I've got a few more slides, and I'm probably not gonna have time to get to all of them. So being a curator of a large collection of pear trees is kind of like opening up a Christmas present when you get the results of an SSR or a SNP analysis. Here's a few clips from a very large dendrogram that Nahla generated. Here on the top left is an angio group. This just shows, and you can see that these are all at the same place on the tree. They don't have little fingers pointing to them, which indicates that these were all identical in the SSR analysis, and not unexpectedly, several different varieties, several different accessions of angio all came out to be identical. But here we see a variety called Sheldon, which is not actually Sheldon. So this identified a mislabeled variety in the collection that was actually an angio. Here on the right, you see a much larger group of things that were exact duplicates of Bartlett. Again, we have sports of Bartlett, which we were not able to differentiate, but there were a few things like Claude Blanchette, which is not supposed to be a synonym of Bartlett. And so that was another mislabeled variety that we were able to identify. Another very useful application of SSR technology is to compare the same variety in different collections to see if we've all got the same thing. And here you can see that the Moonglow pear in the USDA collection is identical to the Moonglow pear in a New Zealand collection. And that was important in the case of Moonglow because they were using this as a standard for some of the DNA techniques. So we weren't able to look at the entire pear collection with the SSRs because that's an ongoing project, but in Sarah's SNP analysis, we looked at a much larger group of accessions And you can see here we have a large group of pears that all came out to be identical to Bartlett, including many more sports and a number of additional things from either old missions in California or some things that were thought to be sports or seedlings of Bartlett, and a couple of more mislabeled plants. The Claude Blanchette that we identified by SSRs also came out by the SNP analysis to be not Bartlett. So I'm going to
We
finish.
have only 2 more minutes before we got to go to Marcus.
Okay, great. Well, I'll just flash through some pictures here. It was interesting that these 4 southern pears, which are hybrid cultivars, these have both Asian pear and European pear in their background. They also had quite a few duplicates. And so I think between about 50 different, I guess 38 different hybrid accessions were identical to one of these, Baldwin, Garber, Kiefer, or LeConte. And here's a group that came out to be identical with Baldwin, and some of these we got from members of the Southern Fruit Fellowship. I think Meany might have been a sampling error because this is a Canadian variety that is likely not identical to Bartlett or to Baldwin. Quite a few identical to Kiefer, including a number of accessions that Maxine Thompson brought back from Pakistan in the 1980s. So apparently Kiefer made its way to Pakistan, where it was well adapted to that warm environment. But then some US pears, like a pear that Tom Burford has been distributing to NAFEX groups, turns out to be either a sport or identical to the Kiefer pear. And here's some photos that I took of some of those different things that were identical to Kiefer in our collection. You can see the size differences might be due to the crop, but these all ripened at the same time and have roughly the same texture and flavor and fruit shape as Kiefer. Here's some more of the Kiefer duplicates. Then here's a group that were identical to Garber. You can read those real quickly. LeConte also had quite a few duplicates, including some that were imported from other countries, Poland, Russia. And here's some photos of the 10 or so accessions in the germplasm collection that were identical to LeConte. So I had some closing thoughts that I was going to share, but we'll save this to the discussion session. I was going to talk about how you folks as citizen breeders can take open pollinated seed from a germplasm collection rather than just looking at a germplasm collection as a source of clones. So thank you for your attention, and I'm going to stop sharing my screen, or Leslie's going to stop my screen from sharing, and we'll turn this over to the next speaker.
Okay, now we will go to Marcus.
Well, hello, everybody.
Maybe I missed my introduction. I'm sorry. Marcus Toole is a home orchardist and chili pepper grower in Georgia. He holds a master's in biology from Georgia Southern University and currently works as the community outreach coordinator for Habitat for Humanity in Bullock County, Georgia. His main growing interests are pears, plums, chili peppers, and citrus. Okay, Marcus, thanks.
Hi. My approach is much more the practical, how this works with the homeowner in a strong fire blight intensity area of the country. If we can get the first slide, that'd be awesome. What you see on the screen there are 3 buckets of LeConte pears from my LeConte pear tree. LeConte pear tree is really the first high quality pear that was one that would do well in Southeast Georgia. It was an accidental hybrid. A guy by the name of John LeConte was in the military and bought what he thought was a Chinese sand pear tree from a nursery in Philadelphia, moved to his home in Liberty County, which is the LeConte plantation, planted a tree, and then when the tree bore pears, they turned out to be pretty high quality soft pears. So this is sort of the real first. This is what made growing pears possible in Georgia, was the introduction of the LeConte pear. In fact, by the late 1800s, the state of Georgia had a thriving pear industry because the LeConte pears in Georgia would ripen before the ones in New York, and they would be shipped by train to New York City and sold, and got a really good price. I have a LeConte pear tree, and I would tell you there's multiple things out there with the LeConte name on it. Not all of them are fire blight resistant, and not all of them have the quality of the original LeConte pear and the one that I have. But I think the one I have is the original, and I can tell you, it's a pretty. I mean, I can't say that it's the best tasting pear, but it is as good as any that you are likely to encounter in a grocery store when it's perfectly ripe. And it's a good seller at the farmer's market as well. Next slide. Before I talk about this pear, I want to talk about some of the other southern pears out there, and I want to talk a little bit about the climate that I'm in. I'm in a little town called Statesboro, Georgia, Bullock County. We are about 50 miles from Savannah. If you're out in the county, your climate is probably zone 8B, but here where I am, I'm basically in downtown Statesboro, so I'm in a little zone 9A kind of climate. My two biggest issues in growing pears, apart from critters, is fire blight, chilling hours. There are actually three, and the other is bacterial leaf spot. Those are the three biggies. And if you're in South Georgia in this kind of climate, you know, a tree isn't going to live very long unless it's extremely fire blight resistant. All of my trees, as far as I know, are grafted onto calorie rootstock. The company that I got my pear trees from, that's what they used. That's what they recommended. And I have seen what pears do if they're on a very resistant hybrid rootstock. And the trees around here that are like that are really huge. I do have two trees on a neighboring property that are 60-foot tall, but for your home gardener, calorie rootstock's pretty good. You end up with a tree that's, you know, maxes out at about 20 feet ordinarily, and that's a decent size for a pear tree in a person's yard. You know, some of the old classic pears that do well here, Kiefer does okay. I don't think Kiefer is as fire blight resistant as some say it is. I've seen an awful lot of it on the Kiefers around. They don't die, but they kind of suffer with it. Baldwin is, I haven't had any problems with fire blight with my Baldwin, but I have a lot of trouble with leaf spot on it. Once again, the tree lives, but the last 2 years it hasn't been very productive because of leaf spot. Another one that if you'd asked me 3 years ago, I would have said this is the best pear in my yard. And it is a really good one and a really reliable, which is Golden Boy, with the problem that it suffers with leaf spot. And I didn't have that problem the first couple of years, but now that I do have that problem, the jury's out whether or not that tree's really going to be productive long term. Another really good one is a new one that is on your screen, and this is Tennessee. This is the first year that the tree is really produced. It's a 5-year-old tree. Tennessee is a accidental seedling. Well, it's a seedling of believed to be a cross between Hosui and Tennessee. Tennessee is the seed parent. It was this is something that comes out of Texas. The fruit, when you eat it, is just like a Asian pear, This is more fire blight resistant than most of them. It's pretty early ripening. Here this year, the fruit started ripening in late June, and the tree was done by mid-July. So on the early side, by standards of Asian pears. But as far as something to take to a farmer's market that you can call an Asian pear and that the customer would think is an Asian pear, tastes like an Asian pear, looks like an Asian pear, has the texture of an Asian pear, this is really good. I have a Korean giant that will probably produce next year, and hopefully that will turn out to be as good as this. But this is a good one for the Deep South. Next slide. This is another relatively new one. It's called Scarlet. Only place I know that has it is Just Fruits and Exotics out of Florida. This pear looks like an apple when it's ripe. It really does. It doesn't really turn red. The name, I think, is based on someone's name rather than the color of the fruit. It has a little bit of a red blush, but it's really more of a yellowish pear. I like it best when it's still a little bit firm. To me, it tastes just like apples. It has a very apple-y kind of flavor, a little tart, just a really nice pear. Probably next year I'll have an idea of how productive it really is. The tree's really just getting going, but flavor-wise is good. I haven't had any trouble with disease with it, and it has seems to have a pretty good, pretty low chilling. And that's something I want to talk to you about a little bit. If you're in a southern climate, fire blight disease resistance is really important. But the other thing that's really important is that those chilling hours. I can count on getting about 500 chilling hours in a year with a cool winter. I can get 600 chilling hours. That's way down from what it was like here 20 years ago. And this is something to think about when you're planting pear trees. If you're just getting started and you're in the Deep South, you're in a warm climate, really look at those chilling hours, investigate it, find out what kind of chilling hours you can count on in your area. Most agricultural universities and colleges have a calculator that you can use, and as well as historic data and you can figure out what you can kind of count on. But the other thing I'm going to say is, given that at least here, we are a good hardiness zone warmer than we were when I was a kid. So if you're planting a tree that's going to live 60 years, you might want to make sure that you're over 100 chilling out. That you can count on getting about 100 more hours of chilling than what the tree requires, because there's no reason to assume that we aren't going to continue to get warmer. So really, even if you're in a region that you may not think you need to worry about chilling hours, maybe you do. Really look at what you can count on and figure that out, especially if you're just beginning, and if you're going to invest in a lot of trees, then do that. Next slide. Or are there any more slides? Maybe there aren't any more. The last one. Yes, this is the sister to Tenazui. This is another seedling from, and this is the pears aren't huge, but these are some of the sweetest pears I have seen. This is one called Southern King. Excellent little pear. Nice color as well. And sells, I mean, people just scarf them up because they're so pretty. The other thing about them is that no issue with fire blight, seems to have a pretty low chilling requirement. I think it's probably around 400 hours. I don't know how far down south it's really been tested yet because it's so new. Now, if you're really worried, if you're like down in Florida, the two pears that are super low chilling are, number one, the Golden Boy. The Golden Boy only seems to need about 200 hours. And the other one that's really low chilling is Hood. And I think Baldwin is originally from the Mobile, Alabama area. So I think that would go into Florida pretty well as well. I don't know that these pears are Tennessee would go that far south. The seed parent is Tennessee, and Tennessee, it really needs 500 hours to bear. I know that Just Fruits and Exotics, the tree that they use for their scion wood is over 20 years old and it has never bloomed. And they're in Wauchula County, Florida. So that gives you an idea of what happens to a pear tree if you're in a climate that's too warm for it. So, you know, these are the photos of some of the ones that just pragmatically work. Things to look at when you're looking at a pear tree for your area. One of the things I would say is talk to people in your area that grow pears and find out what works for them. You know, I've learned more from talking to people who actually grow fruit under the same kind of conditions that I grow fruit from than I have from any book. The books help you get started, but it's the people who are in the area who have just sort of lived with your conditions that can really tell you what's going to work and what's not going to work. And then if you want to try something new, well, then just try with one. Don't buy 40 of them. So anyway, now another thing when talking about pears, just some issues in growing them that I end up encountering is, in addition to fire blight, in addition to the leaf spot, is the issue of magnesium. Now, I'm in a really wet climate compared to much of the country. I'm in a really warm climate because I am fighting various pathogens. I use quite a bit of copper sulfate in my in protecting my trees. I basically do a Bordeaux mix on them. One of the things I found is that kind of leaches the magnesium out of your soil. So with pears, as a home grower, if you're having trouble with your pears not being productive or not growing very fast, especially if you pay attention to them about midsummer and you notice that there's sort of like a yellow ring and a dark ring around the leaves, that's probably a magnesium deficiency. And I have found that when I started just putting some Epsom salts around my pear trees, that made a world of difference in terms of how well they do. And that's just a little easy trick. And as far as nutrient deficiencies, that's the main one I struggle with, and I have to add magnesium every year. And it just is the case with pears. And if you have a tree that's not very productive, the two things I would attribute it to is, one, if it never blooms, then you probably aren't getting enough chilling hours. And if it's just that it's just not doing much of anything, well, then you might have a magnesium deficiency. For the Deep South, I'd say another really important pointer, if you're this far south, and is rootstock matters. And I've planted pear trees that had rootstock other than calorie, and not a single one have done well. I wouldn't necessarily know what the rootstock was. I would just know from from where it came from, that, like they say it's a dwarf tree or something like that. I have not seen a pear that was ever labeled dwarf do well in Statesboro, Georgia. It's not to say there isn't a rootstock out there that won't work here. I'm just saying I've never seen a pear tree that was labeled dwarf do well in this county. And I get people all the time asking me why doesn't my pear tree grow? And they got it from a shopping center, and it had dwarf on the label. I was like, well, even if it is what the labels, what the sign said it was and is an appropriate pear for the Deep South, I'm guessing that the rootstock's not very happy with our climate. It's not liking what it's getting here. So I really do. I would encourage you to stick with the calorie rootstock. The other thing is get your pear trees from a nursery that knows fruit and is getting fruit that is appropriate for your area. Tons of the nurseries, your local nurseries, as well as the big box stores, don't necessarily have the best stuff for your area. They get what they can get from their wholesale suppliers, and that's not always the best stuff depending on where you are. So I would always say, whatever region, go to a nursery that and you can determine they're getting stuff that is appropriate for your region. Because if you're starting with a poor rootstock, you're really going to have trouble.
Okay, Marcus. Well, we need to head into the question and answer. Did you have one final wrap-up thing you wanted to say?
No, I think we're good. We're ready.
Okay, then thank you, Marcus, for your wonderful insights and your beautiful slides. And let's see what we can do with the question and answer. Chris, did you see a good question there for us to start with?
Yeah, there's a lot of good ones, as there always is. Since the topic of fire blight has come up, Dr. Sandy asks, can you suggest fire blight resistant cultivars that do well in the Northeast and produce good fruit? And then here's weaving in one other one from Mark Wolbers. Any genetic insights into cold hardy, zone 3, and short season cultivars? Maybe those can be woven in, maybe they can't.
Right. Joseph, do you want to talk about cultivars here?
Yeah, so fire blight resistant cultivars for the Northeast, there's a lot of them. And I don't know why more aren't available from nurseries, but there has been a long-term breeding program by the USDA in Beltsville. There's been a long-term breeding program in Canada, in Ontario, and there's a whole slew of great cultivars. Check out Cummins Nursery in New York. I'm sure they're carrying some of them. Some of the ones out of Canada, like Harrow Delight, Harrow Sweet, Harvest Queen. They've got some new ones that I haven't tried. Something that's crisp, I think Harrow Crisp. They have Harrow in a lot of their names. Harrow Gold, which I think has a different color. There's a lot of good fire blight resistant things out there. Some of the ones out of Kearneysville, West Virginia. The newest one is Gem that they're actually planting in the Pacific Northwest. There have been a lot of releases from West Virginia that are adapted to the mid-Atlantic coast and New England. Haven't really made it as commercial cultivars, but they would all be great backyard trees. I think things like Potomac. What else? None of the others come to mind. Oh, what was the next question?
The zone.
Genetic insights around zone 3 hardiness.
Yeah, so again, maybe it's not quite zone 3, but the material out of Canada is going to be hardier than the material out of the United States. They've been doing some breeding in Saskatchewan. varieties that I wouldn't grow if you could grow something better, but they'd be adapted to zone 3. Pyrus usuriensis from North China has been one of the parents that they've gotten cold hardiness out of for breeding programs in Saskatchewan, in South Dakota. There was a breeding program years ago. So there's a number of pears out there that are quite cold hardy. I don't know if they go down to zone 3, but St. Lawrence Nurseries in upstate New York offers some pears that they've found growing on old homesteads in New Hampshire, Vermont, Maine. John Bunker might be able to suggest things that they found growing up into Maine. Sorry, no specific recommendations.
Okay. Chris, another question?
Yeah, Eric, here's another one. So Adam Dewson asks a question about he's found it very frustrating that some pears can be quite susceptible to various types of rust: cedar apple rust, quince rust, pear trellis rust, and yet there's not much information about rust. Do you know of any information, he asks, sources that might provide some insight on pear rust resistances, and also any personal experiences?
Nahla, since you were investigating the fire blight, did you investigate any rust stuff, or is that a different thing entirely?
No, I haven't actually seen it.
Yeah, it's a different thing entirely. As you mentioned, Chris, there's a lot of different kinds of rust, and the fungal taxonomists have a hard time differentiating between them. But one of the common things about the different rusts and of both apples and pears is that they have an alternate host, and it usually is an indication that you've got the alternate host nearby. They call it cedar apple rust. I don't know if it's actually the true cedars. It's some of the false cedars here on the West Coast. Some junipers, the what's the cedar that grows down in the southern coast of Oregon. A lot of times, if you've got a problem with rust, you can blame it on if you don't have the alternate host on your property, one of your neighbors probably has an alternate host in their landscape. And so unless you can go cut down the alternate host, which will reduce the inoculum, I can tell you some varieties that are susceptible because those are the ones that we see rust in. The species Pyrus betulifolia that's used as a rootstock for Asian pears, when I walk through the germplasm collection, that's kind of a canary for rust. Quince in general seems to be a lot more susceptible to rust than pears. Some years it's bad and some years it isn't, and I'm not sure if it's the climate. Some people spray copper for rust. There are fungicides, for those of you who are not organic, that can help to control rust. It would be great if we had more information about resistance. We've taken notes in the germplasm collection about when there's a bad rust year, which plants tend not to get it, but those are just anecdotal for a single tree, so it's hard to generalize.
Marcus, do you have bad rust problems down by you?
I've seen rust on LeConte, and I've seen rust on Acres Home, but not a lot. Now, I'm far enough south in Georgia, we don't have a lot of juniper trees, and that's what it would be. It would be eastern red cedar, which is a juniper. And there is one large, very old juniper tree on a property that's across the street from me, and that I promise you that's where that rust is coming from. But it's never been enough of it to really be a problem. It's just you'll see it, I remove it, and there's lots of it on that tree. So which tells me I see a lot of it on that juniper tree, but I don't see it on either LeConte or Acres Home. So while it has some susceptibility to it, I don't think it's real bad.
Okay. All right, Chris, let's go for another question.
Marcus was talking about leaf spot, and I'll second what you were saying, Marcus, that a lot of the varieties that you grow in the South are very susceptible to leaf spot. A lot of people in the North don't even see leaf spot unless they're growing one of the hybrid cultivars, and then it's all over the place. And we see it on a lot of the Asian pears that were probably parents of some of those hybrid cultivars.
Yeah, and I'm hoping, we've had 2 wet years back to back here in Southeast Georgia. And I'm hoping that's the reason for it, and that when we get, especially in the springtime, being wet in the springtime, I think is really sets you up for it. So I'm hoping we'll get a little drier spring, and that will take care of it, at least for a year or two. But we'll see. You know, when you do this stuff, and of course, the other thing, if you have a home orchard, and I'm always, you may, as time goes on and as the pathogens find you, all that stuff just slowly gets worse and worse and worse. So just be aware of that issue. If you're starting out and you go, oh, this is easy, I don't have anything with pears. Just wait, you will.
And just to take off on that, this has come up in some of the other sessions this week. You can just do a lot of cultural things to reduce your disease influence. And one of the biggest things I do in my home orchard is I just prune the trees so that they're open and they have airflow, and you don't get that buildup of moisture that promotes fungal diseases in particular. So leaf spot and scab and mildew and maybe not rust, but those other 3 diseases, just opening up your trees so you have a lot of good airflow. And they would even prune a lot harder than some of the suggestions that have been made in the past by some of the other folks.
Yeah. And I want to mention another, just a really pragmatic thing, especially for a home grower. If you have gray squirrels and your tree's too close to other trees, you almost might as well cut it down. They're going to get every one before they're ripe enough for you to want to eat them. So I mean, and here's basically, unless you're in an area where you can actually have a good way of trapping or killing gray squirrels, it's really important that the squirrels cannot get to your tree from above. If they can get to your tree from above, from another tree, you're done for. But if you can keep your pear trees far enough away from other trees, or keep them small enough that they have to go on the ground and run to the tree, and most pear trees aren't big enough for a squirrel to feel safe in that tree. If you have it where they have to go on the ground, especially if you have a pet like a dog or a cat, that won't stop them from going in and getting a pear, but it will stop them from sitting in your tree all day and eating all the pears. So that's just a little pragmatic thing to FYI if you're planting a pear tree.
Joseph, I understand I'm told that you brought some pears to show us. Can you show us some of those?
Yeah. Actually, I've got a bunch of plates of pears here. Didn't have time in my session, but I'll start. I did pick some pears back in September of some of the varieties that I was going to talk about, and maybe I'll share this with y'all. This is the LeConte that Marcus showed earlier. I've never really had an opportunity to taste a properly ripened LeConte, and these are looking like they probably are ready to eat. Some of those, well, this is a little bit too ripe. You can see the browning in the middle there. Let's try a different one. Too bad we're not meeting in person. We could put these out for you guys to try. I also picked some fruit of the varieties that were the parents of the Old Home by Farmingdale rootstocks. So actually that's not bad. I did want to show you the Old Home pear and the Farmingdale pear, even though the Farmingdale is not actually the parent of the Old Home by Farmingdale. But I'll just confirm by taking a bite of this that you don't want to be eating the Old Home pear.
Very brave of you.
Yeah, it's just mealy and tasteless, and I thought it was going to be more astringent. But I have also got both the Farmingdale. Let's get this closer to the camera, and now we know that Anjo is for sure one of the parents of Farmingdale. So I've got some Anjo here as well. Normally Anjo is a lot bigger than this. So let's see, let's hold the Anjo closer and the Farmingdale further away, so they're about the same size. But you can kind of see the resemblance. Anjo here has short stems. They've got a similar skin, kind of random russet in our climate. Farmingdale, I don't know why people don't grow Farmingdale, and maybe we need to start breeding rootstocks all over again, actually using Farmingdale as one of the parents. Yeah. Now, too bad you can't grow this in the South, Marcus. Farmingdale is really strongly resistant to fire blight, but it probably has a high shelling requirement. And,
Yeah, that would be a show ender. This fire blight.
yeah, and people talk about Farmingdale not being a very good pear, but I think it's just as good as Anjo, which maybe you don't like Anjo to begin with either. Yeah, I kind of like Farmingdale. It's got a little bit of a spicier flavor than. Anjo's the one.
I'm sitting here amazed that you have a LeConte, because here LeConte is finished in July.
Well, we don't get the heat here, and so our LeContes don't ripen until the end of August. These were kind of on the overripe side, but and you can see they're brown in the middle. But I've also got some Kiefers, which for those of you who aren't familiar with the pears that are grown in fire blight prone areas, LeConte is kind of rounded and smooth, whereas Kiefer and Garber both have this distinct kind of a diamond shape. They're wider in the middle rather than wider at the base. And Kiefer can be a good pear. Hedrick in Pears of New York compared it to a raw potato, but if it's not properly ripe, it does have kind of a potato-like texture.
It's a much maligned pear, but I just have to say that story I shared at the beginning, that pear ended up being a Kiefer pear, as you actually helped me identify, Joseph.
Okay, great. So any more questions, Chris? I do have some more pears here I can share, but
pears for your heirs, I guess. Let's see here. Some kind words from the new curator, Lori Reinhold. She says, not a question, but I wanted to say thank you to Eric and Chris for organizing, and thank you to Joseph, Nahla, and Marcus for your wonderful presentations. And thank you, Joseph, for the introduction. I look forward to continuing involvement with NAFEX as the pear curator. I think that's wonderful, Lori. I'm glad that you're on this call, and I look forward to meeting you. I've got some new scion for you guys too.
If we don't have a question right now, I do want to get out another little pointer. Just one of the little pragmatic things if you're growing pears. Some pears are way better than others about not turning brown when you cut into them. Like that's my biggest complaint about LeConte, is that they browns really easily. A really good one for not turning brown is Baldwin, but the Tennessee pear, that's kind of its claim to fame. That's one that doesn't turn brown at all when you cut into it.
I happen to have some Baldwins here, or at least the ones that are labeled Baldwin in the Germplasm collection. Does that look like Baldwin to you?
Doesn't look like mine, but
okay. Well, I'm not so familiar with those pears. We depend on folks like you to tell us if we've got the right thing for some of these. Yeah.
Well, I have what Just Fruits and the Exotic sells, so that's so the question is, where did they get theirs?
Well, we've got a tree labeled Baldwin, and we've got about 6 or 8 other trees with different names that are exactly the same thing. And I'll have to get one of you folks from the South to confirm that it is or isn't.
I'd like to weave in a question. There's actually 2 routes we could go here from that. But I'm going to ask first from Aris, excuse me for mispronouncing your name. How does astringency affect different pear characteristics of pears? Do you see any advantages to breeding more astringent pears? And I suspect that has to do something with the browning that you're talking about.
A couple comments about browning. Most pears, well, there's a difference between browning the cut surface and premature ripening of the center of the fruit. And most pears, unlike apples, pears really need to be picked when they're not quite mature, because they don't ripen evenly on the tree. They ripen from the inside out. So if you leave like a Bartlett and most of the European pears, if you wait until they're ripe on the tree, the center of the fruit will be mush. And so it's not just for storage and shipping that pears are picked when they're almost green, but it's to allow them to ripen evenly. And in fact, a lot of pears need cold storage. Some of the winter pears, they need to be picked when they're just approaching maturity, or else if you ripen them on the tree, they will be brown in the middle. Browning of the cut surface is a different phenomenon. And I don't know, Nahla, if you know more about the physiology of what causes browning. There has been some breeding effort to select for varieties that don't brown as readily when they're cut. You know, the apple folks are real interested in that so that they can serve pre-cut fruit and not have it turn brown before the consumer gets to eat it.
Yeah, I don't know enough about it, but I understood that there is a gene that is known to be responsible for that. And there's been some gene editing work for that, but I'm not very familiar with it though.
Yeah, and that's what's led to those Arctic apples is they've silenced the gene that produces the enzyme that causes that oxidation reaction. I know if you go through a collection, whether it's apples or pears and cut into the fruit, there is a big difference in how quickly they brown. And I think we've done a little bit of that in the pear collection. Looking at browning, nothing comes to mind as being particularly resistant or susceptible. I think they brown more quickly if they're less mature also. So if you've got a fruit that's more ripe, it tends not to brown as quickly.
Here's another one that we could going the other way from what was previously brought up. Janet Flutterer, I'm sorry if I'm mispronouncing your name there too. She said incorrect fruit labels on nursery purchases are always a worry, just like Marcus just brought up. Is there a way for the public to use your genetic analysis?
I think the nurseries are beginning to use the genetic analysis. They didn't really have, you're right, there's a lot of trees that come from nurseries that are incorrect. And as Marcus mentioned, there's a lot of trees that are sold in big box stores that are not adapted to the areas where they're being sold. And it's not just nurseries. We've gotten material in the germplasm collection from private collectors, from other research stations. There's a lot of mislabeled trees out there, pears, apples, quince. But now that these fingerprinting tools are available, I think people are becoming more aware and it's becoming more affordable to try and figure out who's got the right ones and get them into production.
If you do have a question about a pear tree that is very, very important, contact me. We can compare, we can fingerprint it and compare its fingerprints to what we have in our collection already, and then tell you if it matches, you know, things.
Somebody was asking at an earlier session where they can send samples for commercial fingerprinting. There are some labs. I know University of California, Davis does that for a fee. Right.
So see, the question, okay, if you have a specific question about the tree and you have, you know, the question is related to another tree, you want to compare, then you can send both samples. But the issue there, Joseph, is if you don't know what the name of the tree is and you have to compare it, if it's unknown, you want to compare it to an existing database of a whole bunch of pears that you know. So in that case, I don't think anybody else besides us have anything like that. Does that make sense?
The Apple folks have talked about that, and they're a little bit ahead of the pears in developing a collection of reference varieties that have been fingerprinted. But you're right. If we get an unknown and that unknown is not among the ones that we've already fingerprinted, we don't have something to compare it to. But But if it does match something in the collection, we can tell you that it's a match.
Exactly. Yeah.
Okay, Chris, we have time for one more question.
We've got Ed Campbell up on the hot seat, and he asks about anything known about the origins of Warren or Lane pear.
Yeah, Warren pear really stood out in our recent SNP analysis. Of course, Warren pear came from T.O. Warren in, was he in Mississippi?
I think so.
I met Mr. Warren when he came to one of the NAFEX meetings years ago. I've been a member of NAFEX since about 1980, so I remember some of the old timers. I think it was Mississippi, where the experiment station there was evaluating some of the seedling selections that were coming out of the USDA breeding program in Beltsville. And it turns out the Warren is either a sister to, or it might even be identical to the Magnus pear. Nahla, you've got fingerprinting evidence. I don't know if you remember, and I think Sarah's SNP data, I think showed that our Magnus and our Warren pear were identical. They're both fire blight resistant, low chill varieties. I don't know if they'll grow, how far south they'll grow. Warren has.
They don't have enough chilling hours for here.
Yeah. They're liking Warren pear in Western California. A lot of the amateur growers there and some small commercial growers are. One of the problems with Warren is it takes a long time to come into production, but once it starts, makes a nice fruit. It's fire blight resistant. Well, in our last minute, I'm going to hold up one more pear because I've got some white Doyenne, which is one of the founder cultivars of a lot of the ancient pears. And I tasted one of these for the Heirloom Pear Conference just a couple of days ago that Idaho is running. And this is a delicious pear. Alice Waters, one of the famous chefs in Oakland, California, was quoted as saying that this is one of her favorite pears. Oh,
I just checked. Yeah.
you guys should all be jealous because this is delicious. So
You make
sorry
us
I can't
hungry.
share it with you, but the white Doyenne, it's hundreds of years old, and it's a parent of many of. I think when people were planting seeds to develop new varieties, of course they took the best varieties to harvest the seeds, and this must be one that a lot of the early plant breeders collected seeds from.
Yeah.
Okay. Well, on behalf of NAFEX, I would like to thank our panel for making this a great session. Once again, recording of this session will be made available on nafex.org in 24 hours and posted there for about a year before we'll move on to our YouTube channel, NAFEX TV. There should be other downloadable content on our webpage for conference attendees to enjoy. Joseph, is your slides going to be available there?
Yeah, I already sent a PDF of our presentation to Leslie, which she posted. I added a few additional slides, so I'll have to send her an updated copy. I also sent her a paper that we wrote about the Old Home Farmingdale story, which has a lot more detail about the origin of those two cultivars and why they were considered important parents for breeding. So check out that paper from.
Great. Looking forward to that. Okay. Well, don't miss out on other sessions in our conference, which is not over yet. We've got another one this evening and 2 more tomorrow. So stay connected on social media and our website, and you can find all the links at nafex.org on our homepage. Thank you.
Thank you all. Thank
Thanks,
you.
Eric. Thanks, Chris. Thanks, Marcus.
Thank you.
Thanks.
Bye-bye.
Bye.
Bye-bye.